Okay, we pick up this discussion with a look at the development of the reproductive system. And what we need to appreciate here is that early in development, primitive structures that could produce either the male or female duct system as well as the gonads are present. And if we follow the default pathway of the development of the male duct system, what we see is a degeneration of the paramesonephric ducts while the mesonephric ducts persist and become closely associated with what will eventually be the testes, which will drop, guided by the gubernaculum, through the inguinal canal into the scrotal sac. And you can see the other adult structures that develop: the seminal vesicle, the prostate, the vas deferens, the epididymis, and of course the male urethra, the bulbourethral glands, all present. Notice that the mesonephros eventually vanish, okay, with only the gonadal ridges remaining. Now, if we follow the female developmental sequence, what we see is the paramesonephric ducts persisting. The gonadal ridges will become the ovaries, and then, of course, the fallopian tubes and uterus, along with the vagina, will develop from these primitive structures. This is a look at the development of the external genitalia, and if we start at the top, what we have are indeterminate external genitalia that become sexed as a result either of an absence or a presence of a special gene product that is encoded by the SRY gene, along with, of course, the presence in the male of increased levels of testosterone. At 5 weeks post fertilization, the genital tubercle folds and labioscrotal swellings are identical in the male and female. There's really no difference. But by 10 weeks of development, as you can see down here, the male penile structures are larger than the clitoris, as we would see here when we compare the male and the female, but the urethral groove, you can see down here, is still open in both the male and female external genitalia. Near term, what happens is that the adult condition will emerge. The labioscrotal swellings become the scrotum in the male and the labia majora in the female. The genital folds fuse in the male to form the body of the penis, where they form the labia minora in the female. Okay, so you can see here that we end up with a raphe in this case as we close at midline. Here, the vestibule requires that the raphe remain open. The urethral groove in the male is enclosed by the raphe of the penis, but it remains open in the vestibule of the female. You can also see that the glands of the penis, along with the corpus spongiosa cavernosa, are embodied in a female counterpart, the clitoris, which is found anterior aspect of the vestibule, enclosed as the male reproductive organ is in a prepuce covering the glands. At 60 days, the embryo becomes a fetus. The fetal period lasts from this time until birth, and here we see rapid growth of body systems and overall body dimensions. We see the emergence of lanugo, which is a fine, soft covering of hair on most of the structures in the fetus, and there's vernix, which is a waxy protective coat that also aids again in essentially protecting the fetus from damage inside the womb. We also note that throughout this time it's been floating in the amniotic fluid in the amniotic sac, which allows freedom of movement but also damage from physical force. Parturition is essentially birth. In the mother, estrogen overcomes the inhibitory influence of progesterone, and what happens as a result is that the uterus becomes more excitable, more likely to generate action potentials that will hit the brain and result in the release of oxytocin, which will promote uterine contractions. And what this will do is generate a positive feedback loop whereby the stretching of the uterus uh, produces action potentials that influence the brain at the hypothalamic level to release oxytocin, that in turn spurs more uterine contractions, and eventually the baby is out. As you can see here in the second stage, the baby emerges, and in the third stage, the afterbirth is expelled. What brings this on is that in the fetus, the adrenal gland is getting larger before parturition because of the development of the fetal hypothalamus. Remember that the hypothalamus has a direct impact on the pituitary gland, causing it to release adrenocorticotropic hormone, which will in turn target the adrenal cortex, resulting in the release of corticosteroids. And it's these corticosteroids which will change now the sensitivity of the uterus to the inhibitory effects of progesterone. Labor has three stages. The first stage is the dilatation of the cervix and the rupture of the amniotic sac. In the second stage, at maximum dilation, the baby is going to exit the vagina. And then in the third stage, we see expulsion of the placenta from the uterus. If we look here at the complex hormonal interaction between the endocrine system of the baby and the mother, what we see is that what spurs birth is the fetal hypothalamus secreting corticotropin releasing hormone, spurring now ACTH secretion from the pituitary, which is going to target now the adrenal cortex of the developing fetus. What happens as a result is that the fetal adrenal glands generate large amounts of corticosteroids, which travel in the umbilicus to the placenta. And here, the adrenal corticosteroids cause progesterone synthesis to flatten, and estrogen and prostaglandin synthesis increases. The uterus, as a result, becomes more excitable. As it stretches, the action potentials impinge on the mother's hypothalamus, resulting now in the release of oxytocin from the posterior pituitary, and that brings about the contraction of smooth muscle in the uterus that ultimately results in the expulsion of the baby and the afterbirth. And remember, it's that positive feedback loop that will persist until the baby is completely out of the birth canal. The stretching of the uterus stops, the impulses that get sent back to the hypothalamus cease, and the production of oxytocin drops. Now, what we're going to look at next are some selected body systems in the newborn. We'll take a peek at the respiratory and cardiovascular systems. We've talked a little bit about the digestive system already. If we look at the respiratory and cardiovascular system, what we have to appreciate is there's a particular challenge in the newborn. We have to get oxygen both to the pulmonary tissue as well as to the rest of the body. And in order to do that, we have to put into place some shunts that allow oxygenated blood to return from the placenta and nourish these tissues in the developing fetus. The foraminal valley is one of the bypasses that exists. Its purpose is to allow oxygenated blood to move from the right to the left atrium, while the ductus arteriosus is a bypass between the pulmonary trunk and aorta that again allows oxygenated blood to fill both of those great vessels. The pulmonary trunk, of course, bringing, in this case, oxygenated blood to the lungs in the fetal circulation, while the aorta, as we already know, brings oxygenated blood to the rest of the body structures, so that takes care of the systemic circulation. The umbilical vein and the arteries are present to bring oxygenated blood back from the placenta, and eventually what will happen is that this will pass under the liver through the ductus venosus and will enter the inferior vena cava. and so oxygenated blood will flow from the inferior vena cava back up to the right atrium of the heart, where those two bypasses, the foramen ovale and the ductus arteriosus, will allow blood to be shunted both to the lungs and also into the aorta and to the rest of the body. What happens at birth is that all of these bypasses will close. The ductus arteriosus will shut between the right and the left atria. In addition, the umbilical vein and the arteries will degenerate into ligaments, um, just as the ductus venosus and arteriosus will. The result then is that we don't have any patent fetal circulatory structures in the neonate, and the adult circulatory pattern will predominate. What happens in the digestive system? Well, sometimes we see the accumulation of meconium, which is a mix of cells from the GI as well as the amniotic fluid, bile, and mucus that are excreted by the newborn. The stomach begins production of acid. The liver does not produce bilirubin for the first 2 weeks in the newborn. Lactose can be digested, but other food has to be gradually introduced. The reason for this is that there's a considerable amount of lactase present in the GI of the newborn, and that's to handle, again, the disaccharide lactose, which is present in both colostrum and in breast milk. What we're looking at here are just the circulatory changes that we've been discussing. Prior to birth, we have the fetal circulatory pattern. You can see that blood bypasses the lungs by flowing from the pulmonary trunk through the ductus arteriosus and into the aorta. It also bypasses the lungs by flowing from the right to the left atrium through the foramen ovale, and bypasses the liver by flowing through the ductus venosus and eventually entering the inferior vena cava. Oxygenated blood is returned to the fetus from the placenta by the umbilical vein, while deoxygenated blood is carried from the fetus to the placenta through the umbilical arteries. And so what you see here essentially is a way to get oxygenated blood to all tissues. The lungs at this point are useless as respiratory organs because remember that the baby's inside the amniotic sac and the lungs are filled with fluid, and as a result do not engage in any significant amount of gas exchange. But we still have to provide oxygen to the developing lung tissue, and that is accomplished by bringing oxygenated blood into the right atrium, from there to the right ventricle, and from the right ventricle we enter now the pulmonary circulation, bringing oxygenated blood to the lungs. In addition, as we've already seen, blood is shunted from the right to the left atrium. The left atrium is going to force blood into the left ventricle. The left ventricle forces blood into the pulmonary trunk. And remember that the pulmonary trunk in this case, the left ventricle forces blood into the aorta, which is joined to the pulmonary trunk in this case. As a result, the great vessels can share oxygenated blood between them. This ensures that we deliver blood with a reasonably high oxygen content to all tissues, allowing them to get the oxygen and nutrients they need to fully develop. At birth, we see quite a few changes. When air enters the lungs, blood is forced through the pulmonary arteries to the lungs. The ductus arteriosus closes and turns into the ligamentum arteriosum. The foramen ovale will shut, becoming the fossa ovalis, and so blood can no longer flow from the right to the left atrium. The ductus venosus collapses and becomes the ligamentum venosum, running on the underside of the liver, and the umbilical arteries and veins, once they're cut, will become the round ligament of the liver, and you can pick that up down here, the round ligament, and you can also see here the ligamentum venosum, the remnant of the ductus venosus. The umbilical arteries also degenerate and become the cords of the umbilical arteries. So you can see here that once we're out as the neonate breathing air, it's primarily pressure changes that bring about the collapse of these bypasses and restore now the normal blood flow um, in the neonatal heart. I mean, we don't need these fetal structures. I should point out that occasionally the fetal structures will persist. In this case, we call them patent structures, and in most cases, this results in a decrease in cardiac output of oxygenated blood and can often result in a failure to thrive. So surgically, we have to intervene in order um, to restore normal blood flow in the neonate and as a result provide oxygenated blood to all tissues. Once born, one of the ways that we assess whether or not the baby is developed and will manage to thrive is through the use of something called an APGAR score. APGAR is an acronym standing for Appearance, Pulse, Grimace, Activity, and Respiratory Effect. And it's rated on a scale of 0 to 2, where 2 denotes normal function. The total APGAR score is the sum for the 5 characteristics. And so you can see here the different rankings. An APGAR score of 2 indicates a pink skin color, greater than 100 beats per minute pulse rate. We see facial grimaces and the extremities move, and we see active movement and good muscle tone and good regular breathing and a strong cry. We'll score low if any of these are not up to what we would consider healthy standards. Okay, lactation is the production of milk by the breast tissue. This is critical for the nourishment of the neonate. Stimulation of the nipple by the baby's suckling produces action potentials and sensory neurons that connect with the hypothalamus, which causes the posterior pituitary to release oxytocin, while the anterior pituitary generates prolactin. Prolactin stimulates milk production, while oxytocin stimulates milk ejection. And the result then is that as the baby nurses or as the baby cries, what we see is the availability of nourishment. Now, as we head towards adulthood and old age, quite a few changes take place in tissues and organ systems. Cells like the liver and the skin continue to proliferate throughout life, but neurons cease to divide once they've reached a certain number, and the dead cells are not replaced. So after reaching a peak, neurons die first at a rapid rate and then slower as we age. But we do begin to lose neurons and neuronal connection. This is going to have a major impact on the function of the central peripheral nervous system. Mitochondrial DNA function declines. As a result, we have an impact on energy production in the cells. More and more cross-linking takes place between collagen, making tissues rigid and less elastic. We have difficulty in near vision, a condition we call presbyopia, which is a result of loss of lens flexibility. Joints, blood vessels, kidneys, lungs, and heart lose function as cells die and tissue function diminishes. The total number of skeletal muscle and cardiac muscle fibers declines, and the strength of skeletal muscle, which peaks at 20 to 30, begins to decline after that point. The decline in cardiac function causes lower cardiac output and less oxygen and nutrients reach the cells, and obviously this is going to have an impact on cell viability. Arteriosclerosis results from deposits that harden the vessels, and this results in the onset of hypertension. What happens as a result is that blood flow through these regions will slow down, blood pressure will go up, and we run increased risk of generating an embolus, which could jam in a capillary bed or in a narrow diameter vessel and produce a thrombus. And this will result in the cutting off of blood downstream tissues. They will then infarct and die. Aging of the cells happens due to exposure to toxins. Free radicals build up. These are electron-poor species that steal electrons from covalent bonds of viable molecules, and this results in cellular dysfunction, can lead to conditions such as cancer or apoptosis, and a poor diet can result in loss of fuel and reduction in vitamin content, which is going to decrease ATP production. The loss of energy is going to accelerate cell death and tissue death. The immune system gets less responsive to antigens but more responsive to self-antigens, so we see an increase in infection and disease as well as a concurrent increase in autoimmune conditions. Progeria is a condition that accelerates all of these processes, and it's the result of a defective nuclear protein that causes genetic material to degrade earlier than it otherwise would. We now know, or at least we have a pretty good suspicion, that what brings about a lot of the changes that we associate with aging and death is loss of genetic material in the nucleus of the cell through subsequent cell divisions. This is due to the fact that the enzymes that maintain the tips of the chromosomes, which are called telomerases, cannot keep up or at least maintain the genetic information, and as a result the chromosomes shorten with each cell division. And once we get into areas of the DNA that contain critical information, very often the cells will apoptose. This will result in cell death and tissue death and loss of organ and tissue function. Our ability to adjust to stress also declines due to the fact, again, that we've lost nervous and endocrine function, and as a result the body finds it more difficult to maintain homeostasis. Okay, we're going to take a little bit of a left turn here, and we're going to discuss what's really at the core of all of these processes that comprise human development, and that's the study of genetics, which is essentially the passing down through generations of information carried on the DNA molecule that is responsible for the appearance, behavior, and the function of every tissue in our body. We pass these traits to our offspring during the process of fertilization. Mendelian genetics essentially describes how these characteristics are passed down. There's some terms that we need to become familiar with. When we're talking about an individual's genotype, we're referring to the actual alleles that the person possesses for a given trait. Now, what is an allele? It's a hunk of genetic information, a gene, that will determine generally a protein structure that's responsible for a particular characteristic. The phenotype is the physical manifestation of that characteristic. Many traits, not all by any stretch, but many traits are inherited via dominant and recessive alleles. And essentially what we mean by a recessive allele is that in order for the trait of the recessive allele to appear in the individual that inherits it, they have to have two copies of that recessive gene, one from the mother and one from the father, in order for the characteristic to show up. Whereas for the dominant allele, you only need one copy of the gene for the characteristic to appear. Sex-linked traits simply reside on the 23rd pair of chromosomes, the sex chromosomes, and they are found primarily on the X chromosome. Um, because the X chromosome contains information that relates not just to the development of the um, the reproductive system, but also body plan information. Many traits that affect other body systems are sex linked as a result found on the X chromosome. What we what we're going to discover as we continue our discussion of genetics is that these sex-linked traits are going to be more prevalent in males than in females because men are hemizygous for all X-linked traits because they only have one X chromosome, whereas the female has two. And so for a male to manifest either a recessive or a dominant sex-linked trait, they only need one copy in either case, whereas for a female to express a sex-linked trait, um, it, it would follow um, the same inheritance pattern that we would see in an autosomal dominant and recessive alleles, except here we're just looking at the sex chromosomes. So for the female to express a sex-linked trait that's dominant, she would only need one copy of that allele, but to express a sex-linked trait that's recessive, she would need two copies of that allele, one on each of her X chromosomes. And this is why sex-linked recessive traits are much more frequent in males than in females. Homozygosity simply means having two of the same allele for a trait on each chromosome, both your maternal and your fraternal chromosome. An example would be a homozygous dominant individual if we designate, let's say, an allele for skin color, the albinism allele. The recessive condition would be written as a lowercase a, and the dominant allele would be written as an uppercase A. So if an individual is homozygous dominant, we would describe their genotype as capital A, and you can see that here in the diagram. Let's go ahead and show you that now. We'll hit this in blue. Okay, so you can see here the homozygous dominant condition. All right, this is representing the allele on each of the two chromosomes, and then homozygous recessive would be indicated as lowercase a. An individual who's heterozygous will have one of each type of allele, and if this allele were inherited as simple dominant recessive, this particular individual would show the dominant trait because the uppercase allele would mask the influence of the lowercase allele. And so here you see an example of an individual with albinism. Okay, this would be their genotype. And then an individual with normal pigmentation could be either homozygous dominant or potentially could be heterozygous. Okay, either would produce the phenotype seen in the picture of darkly pigmented skin. Now, chromosomes are the DNA molecules that carry the pieces of information that dictate all of the traits that we associate with the human condition. Genetics is the study of heredity. DNA is the hereditary material found in the nucleus that controls virtually everything that the cell does. The chromosomes are numbered in order from biggest to smallest. So chromosome number 1 is the largest, and chromosome number 23, or 22 rather, is the smallest. The sex chromosomes you can see here are quite different in size, the Y chromosome being rather small, the X chromosome being quite large. We determine an individual's chromosomal content using a technique called karyotyping, where we take a tissue sample and then we freeze the cells at metaphase of mitosis, rupture the nucleus, and then we stain the chromosomes and look at the banding pattern, take a photograph of that, and then we do a little cut and paste and arrange the chromosomes by banding pattern and do a count. The condition in which we have 23 pairs of chromosomes is called euploidy, meaning the normal chromosome number, whereas if we have more or fewer chromosomes than 23 pair, we call that an aneuploid condition, and that's generally associated with defects in body systems. We are diploid organisms, so we have two copies of our chromosomes. We get one copy from our mother and one from our father, and we do that as the result of the fusion of haploid gametes, the egg bringing its 23 chromosomes, the sperm bringing its 23 chromosomes, for a total of 46. And at that point, we've met the conditions for generating a unique human life. The karyotyping that we see here in this photograph is an example of using the technique of freezing the cells at metaphase of mitosis, rupturing the nuclei, staining the chromosomes, and then doing a count. Okay, the prefix karyo means of or pertaining to the nucleus. We often do this before the child is born in a process called amniocentesis, and what we are able to determine from karyotyping cells that are sloughed into the amniotic fluid is the genetic content of the body cells of the developing fetus, if we're talking about amniocentesis, or the embryo if we're talking about a technique called chorionic villus sampling. And that can inform the parents what steps to take in the event that we detect an aneuploidy. Now, we should also point out that we can use karyotyping for another very important technique, which is the determination of the presence or the absence of defective genes. And this process is called PCR, polymerase chain reaction. What we do in PCR is we isolate genetic material from a sample. In this case, let's imagine that we're doing it from cells isolated from amniotic fluid. And then if we know, as a result of family history, that there are single gene diseases that run in the family via the prospective mother or father, we can determine whether or not the baby carries those conditions by designing DNA primers that flank the gene suspected in that condition. We can amplify that genetic material, and then we can sequence it, and we can determine whether or not the child carries potentially a disease allele. So we can do much more with amniocentesis and CVS than before the advent of PCR. Okay, homologous pairs of chromosomes are essentially look-alike chromosomes, one from the mother and one from the father. A locus is the physical location of a gene on a chromosome. We generally define loci as existing on the long or the short arm of a particular chromosome, and a lot of that has to do with our knowledge of the banding pattern. We can even determine the location of a gene on not just a chromosomal arm but a chromosomal band. And now that we've sequenced the entire human genome, we can even determine reasonably accurately the number of base pairs away from the centromere or the telomere that a particular gene locus is located. Remember that alleles are different forms of the same gene. Many genes have multiple alleles, which means that there are going to be flavors of instructions for a particular protein that does a job in the cell, some of which may be dominant, some of which may be recessive, or in some cases you'll have a condition known as codominance, where if you have one of each type of an allele, you'll see physical characteristics of both. Okay, an example of that would be the alleles for type A and type B blood. If an individual has one allele of each, their blood is considered AB, okay, because you can detect both gene products, the results of both gene products on the surfaces of red blood cells. Okay, so that's another type of variation on inheritance. You can have what we call codominance. And so here are again some examples of um, gene dominance. Complete dominance is, as we described earlier, um, if the dominant allele is present in one or two copies, then only that trait is going to present. In codominance, if you have one of each type of an allele, you will see physical manifestations of a trait from both alleles. And in incomplete dominance, the dominant and recessive allele are both expressed with the recessive generally at a much lower level. Okay, so that's another variation on inheritance pattern. But it turns out that most traits that we think of when we think of human characteristics are not inherited in any of these three patterns. Most traits are polygenic, meaning more than one gene is going to have an influence on the manifestation of that physical characteristic. And so examples of this include intelligence, height, weight, and eye and skin color, just to name a few. As a result, you don't get simple mathematical ratios for distributions in live births when we're talking about polygenic traits. Instead, what you generally see is a normal distribution, a bell curve, where you have very few of the extreme manifestations of a particular trait, and the bulk of live births exist in the peak of the bell curve. Sex-linked traits are those carried on the sex chromosomes. Most sex-linked traits are carried on the X chromosome due to its size and due to the fact that it contains body plan information. And we can tell this because there are defective genes on the X that affect other body systems aside from the reproductive. As an example, examples of some X-linked traits include hemophilia, the bleeder's disease, color blindness, and also muscular dystrophy. X-linked traits more often affect males because the Y chromosome doesn't have any homology to the X chromosome. So any information that's on the X chromosome in the male is going to manifest itself. The male is hemizygous for all X-linked traits. There's only one trait that we've identified on the Y chromosome, and that's the trait of being male. The SRY gene is thought to be the locus here, and it is responsible for the emergence of the testes, the male gonad, and also, as we find out, the production of the male external genitalia duct system. Okay, meiosis is the process of the production of haploid gametes from diploid stem cells. In meiosis, what happens is that homologous pairs will synapse and exchange genetic information, and then 2 rounds of cell division will result in the production of haploid cells from diploid precursors. These gametes will unite in fertilization to restore the diploid condition and meet the conditions as a result for a unique human being. Here, homologous pairs of DNA are reunited. As a result, you're a mixture of genetic material from both your mother and your father. And as we're going to find out in a minute, in the production of these gametes, of these sperm and eggs, we actually take the order of genes on chromosomes, and we swap genetic material between homologous pairs, resulting in unique gene combinations within a gamete that don't necessarily exist on the chromosomes of the diploid cells of either the mother and the father. As a result, we increase genetic diversity by the exchange of genetic material and also by the process of amphimixis after fertilization, resulting in unique combinations of genes that obviously are not present in either the mother or the father. So let's take a look at how meiosis works.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having two sets of chromosomes, but will undergo meiosis to produce haploid cells, having one set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of two identical strands of DNA. Meiosis involves two divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, two homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only one of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Okay, let's touch a little bit on some genetic disorders. These are changes in the sequence of DNA that result in an alteration of physical or physiological characteristics. Congenital disorders are birth defects that are not necessarily genetic. They can be brought about by changes in the environment within the uterus. As an example, teratogens, which are compounds that mutate DNA and affect development, can produce abnormalities in morphology. An example of a teratogen is thalidomide. Um, Another type of teratogen would be exposure to radiation, exposure to certain viruses. All of these can produce changes in normal human development. Mutation is simply the change of a gene's sequence as a result of exposure to a mutagenic agent. A mutagenic agent could be a chemical, could be radiation, could be viruses. It also occurs as a result of the DNA replication machinery itself. There is always going to be an error rate in any large number of mitotic divisions, and if these mutations lead to a change in cell behavior and appearance, often these changes will be intercepted and wiped out. Those cells will be destroyed by the immune system before they get a chance to produce conditions such as cancer. Um, Cancer is loosely described as a disease of uncontrolled cell division um, that results generally from changes in the genetic material. Sometimes it can result from um, cell division signals that come from other tissue locations. Oncogenes are genes that are associated with cancer. Example of these include uh, genes such as SARC or MYC, which were originally discovered and characterized on viruses, but we've now found their cellular counterparts. And when they're overexpressed or when they're mutated, they can produce cells that divide in uncontrolled fashion. Tumor suppressor genes are thought to be gene products that prevent the uncontrolled growth of cells, and it's thought that mutations in tumor suppressor genes are um, one of the mutation events that can bring about a cancerous change. A carcinogen is simply a chemical that promotes the production of cancer cells. Um, Many carcinogens are also mutagens. Obviously, if you have the ability to change DNA structure, you have the potential to change genes that are associated with the control of cell division, and that can produce oncogenic tissue. Um, there are also inherited um, gene mutations or genetic alleles that can lead to a predisposition for certain types of cancer. Um, Examples include the gene responsible for retinoblastoma, the RB gene, and the gene, one of the genes that we've found that are associated with breast cancer, the BRCA1 and 2 genes. Um, but generally, it's not as simple as finding a mutated gene and saying that this is what produces the cancer. Cancer is probably a much more complex series of events that involves both environmental impacts on the control of cell division, as well as changes in the genetic material, as well as um, chemical signals that induce cells to divide in an uncontrolled fashion. So one of the mistakes I think that's often made in the discussion of cancer is to consider cancer a disease when it's actually more aptly put as a symptom. The symptom is uncontrolled cell division. The cause can be any of a number of different origins. And so once we're able to deal with the cause of uncontrolled cell growth, we can then begin to figure out how to control uncontrolled cell growth. What we're looking at here is just an example of a typical um, distribution of phenotypes and genotypes in a simple dominant recessive allele. So here we're taking the example of albinism. What we have to consider now is the genotype of the mother and the father. In this example, if we know that the mother is a carrier for albinism, she would have normal pigmentation. Her phenotype would be skin that has normal production of melanin, and she marries a carrier father who would also appear normal. The phenotypic distribution of normally pigmented children to albino children over a large number of births would be 3 to 1, as you can see by this process known as a Punnett square, where we produce the possible eggs from the female on one axis of the square and the possible sperm produced by the male on another axis. Um, the, the genotypic ratio would be 1 to 2 to 1, and that's going to reflect now again over a large number of births the percentage of children we would expect to be homozygous dominant, homozygous recessive, and heterozygous. Now the last panel here, last table, is going to talk a little bit about some of the different disorders and a description of the systems that they impact. Generally speaking, okay, when we look at these different genetic conditions, a dominant condition, a dominant disease, is going to affect a structural gene of some kind. And the reason behind that is that if you think in terms of defective structural components for a tissue or for a cell, if you have some defective structural components, you're going to see an effect on the overall structure. It would be as if you were taking the example of putting together a building using 50% good bricks and 50% bad bricks. Well, despite the presence of the good bricks, the bad bricks are going to produce a structural fault in the building, and as a result, even though not every brick is a bad brick, the building is still going to be structurally unsound. And you can extend that analogy to human tissue. If you have a certain percentage of bad building blocks, the result is that the entire structure is in all likelihood going to be affected. And so this is why dominant disease conditions generally affect structural genes. For recessive diseases, we're generally talking about enzymes that are part of a multi-step pathway. And if you lack the enzyme for a multi-step pathway completely, then the pathway won't work and the disease condition will be realized. If you have production of some enzyme for that particular pathway, let's say that you have one functioning allele and one non-functioning allele, the likelihood is that the pathway is going to run, and as a result, you'll appear normal for that condition. So when you look at some of these conditions in this table, just take special note of that. Also, pay special attention to the sex-linked traits. Note that the majority of these lie on the X chromosome, and as a result are going to more often affect males. Now, female can show an X-linked recessive trait, but it's going to be exceedingly rare that this happens. This is why you very seldom see, for instance, female hemophiliacs. Okay, this is just a look at the consequences of different types of mutation. Here we're looking at addition and deletion mutations.
The nucleotide sequence in DNA determines the nucleotide sequence in messenger RNA and, consequently, the sequence of amino acids in a protein. A mutation in the DNA can result in a change in the amino acid sequence of a protein. One type of mutation that can occur either spontaneously or as the result of a mutagen is the addition of one or more nucleotides during DNA replication. Because translation of a gene begins with a specific codon and proceeds one codon at a time, the addition of an extra nucleotide shifts the codons in the mRNA. This type of mutation is termed a frame shift mutation. A frame shift affects all amino acids incorporated beyond the original site at which the addition occurred. If the new codon generated by the frame shift is a stop codon, the protein synthesized will be shortened and is often non-functional. Another type of frame shift mutation occurs when a nucleotide is deleted during DNA replication. Deletion of a nucleotide in DNA results in a change in the codons in messenger RNA from the point of the deletion and changes in the amino acids inserted into the protein.
And in this final panel, we're looking at mutation by base substitution. So what's happening here is we're changing one base to another. This can result from exposure to a chemical mutagen or to radiation or to a problem in the repair of DNA if it's been physically damaged. So let's take a listen.
A mutation occurs by base substitution when an incorrect base is incorporated into DNA. Some base substitutions occur because purines and pyrimidines exist in two structural forms. The most common form results in base pairing between adenine and thymine and between guanine and cytosine. However, the hydrogen atoms can move to form a base with altered hydrogen bonding properties, creating a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can bond to cytosine. A tautomeric shift in thymine allows it to bond to guanine. This error in DNA replication is passed on to the cell's progeny. The change in a single nucleotide in the DNA results in a change in the corresponding nucleotide in messenger RNA. The change in the codon can result in a different amino acid being incorporated into the protein.